Our first speaker today is Claudia Ancillotti from Biochemie Lab. Biochemie Lab is a large company with a long history of expertise in food and environmental testing. Claudia is the method development manager and will be presenting on the analysis of vet drugs from food. Biochemie Lab is a longtime user of the 7500 platform and is one of the early adopters of the new 7500 plus system. So those looking for performance information on this system will find this presentation very informative.
Good morning to everyone. I am Claudia Ancillotti, the Method Development Manager at Biochemie Lab. I am very happy to be here today, and I am talking about the veterinary drug analysis in meat supply chain using the 7500 system. So, first of all, a short presentation of my company. Biochemie Lab is a young and dynamic company, and it's founded in 1996 by the Bonciani family, and the company has developed over the last 20 years with the contribution of the skills and the professionalism of the entire family. Here we have some of our service, sampling, logistic, environmental, food and food contact material analysis. And we have some of our numbers. The company has 5 sites. The main headquarters is and laboratory is in Campi Bisenzio near Florence. And the company has 25 years of presence on market and a forecast for this year of more than 155,000 samples processed by more than 150 employees. Going to the presentation topic, let me start giving you some background information why veterinary drug analysis is a hot topic. First of all, because the use of veterinary drug is regulated. In fact, the European regulation 37 classified the veterinary drugs in allowed substances for which a maximum limit can be established, and the prohibited substances for which the use of this drug is totally prohibited. Antibiotics are classified as allowed substances, and they are regulated by the establishment of a maximum residue limit. In this slide, I report some example for some of the investigated antibiotics. We can see that the MRL, so the maximum limit of concentration that we can have, is a function of the investigated analyte, but also of the target tissue. So we can have very different concentration if we analyze different metrics. Then the growing attention on the effect of food on health and the possible adverse effect due to the presence of veterinary drugs in meat lead to the growing development of the antibiotic-free supply chain, and this lead to the request of lower and lower limit of quantification, and so to the necessity to develop a very sensitive analytical method. But who are antibiotics? Compounds belong to very different classes, such as those reported in this slide, are classified as antibiotics, and we can see that these compounds are characterized by very different dimension and functional group, and these result in very different chemical properties, so such as polarity, solubility, and chelating properties. And the simultaneous determination of this compound, characterized by very different chemical properties, is the first analytical challenge in the analysis of antibiotics. The second difficult is represented by matrix. In fact, the meat supply chain include very different matrices, from the simplest animal water to the more complex meat, animal feed, biological fluid, and offal. And these metrics were characterized by different interfering compounds. So, for example, salts, fats, protein, and phospholipids. So they are very complex metrics. So how we can perform this kind of analysis? We need LCMS because liquid chromatography coupled to mass spectrometry give sensitivity and selectivity that we need. And in this slide, I report the experimental condition. On the left, there are the chromatographic condition, mobile phase, chromatographic column, and so on. And on the right, we have the MRM transition that we apply in our method when we optimize the condition. So we can see the results, and we can see the extract ion chromatogram of all the analyzed antibiotics in a standard solution and 10 ppb. And below, we have the extra ion chromatogram of a class of this compound that are beta-lactams. In this case, we can observe that the analytes are characterized by very different functional group, and these result in very different chromatographic behavior. So we have dilution in different part of the chromatogram. And the same things happen for the other class of antibiotics, quinolones, macrolides, sulfamides, and tetracyclines. But the importance of using LCMS technique is more evident when we pass from standard to metrics, because we have talked about very complex metrics, and we need the two main quality of LCMS technique that are: the first is selectivity, and selectivity is given first of all by the chromatographic separation of the analyte from the isobar interferences, and the separation of the analyte from the matrix that can cause a signal suppression. Then selectivity is also given by the um, the mass spectrometer, because the use of the MRM experiment allow to analyze each precursor ion with its selected fragment. Then we need also the sensitivity, because the use of a very sensitive mass spectrometer, such as the 7500, is allow to perform sample dilution, so allow to reduce the matrix load. So let's focus from standard to matrix, and we can see some examples: sulfonamide in red meat extract and quinolones in liver extract. And in spite of very complex metrics and a low concentration, we are talking about 0.2 ppb, we can observe a good peak shape, good sensitivity, and a good separation of the analyte from the interferences. But in spite of this, we observe, when we pass from standard to metrics, a different response of our analyte. So the response factor of all the analyte change, and in some case we can have the reverse of response factor. For example, is the case of ampicillin and cefazolin that I reported in the slide. For this reason, we study the signal suppression through the recovery evaluation using two different calibration curves. The first is the standard calibration curve, and in this case, the recovery that we observe are made by two factors: the matrix effect and the real recovery deriving from the extraction procedure. In the second case, we use matrix-matched calibration curve that allow to have the recovery that is the real recovery deriving from extraction procedure. So, as we can see from the gray bar, if we use a standard calibration curve, a lot of analyte have a signal suppression. But only if we use matrix-matched calibration curve that we can see from the red bar, we have very good recovery. For this reason, we choose to use the matrix matched calibration curve as a calibration approach, and I report in the slide some example. We can observe a very good linearity for all the analyzed compound. So we have optimized the LCMS method, and then we have chosen the right calibration approach. Then we can validate our method through the analysis of 6 spiked sample at 2 different concentration level in order to evaluate the method recovery and repeatability, and in order to calculate the method uncertainty. The uncertainty is calculated by the metrological approach as the sum of several contributions. And as we can see in the slide, the main contribution to the total uncertainty is given by the calibration, repeatability, and reference materials. Here I reported the method validation results for white meat, and we can observe that almost all the analytes show a recovery in the range 70-120%, and only a small number of compounds show a recovery in the larger range 30-140%. So, as recommended by the European SANTE guidelines. Finally, I report the proficiency test results at which we have participated, and we can observe some of the analytes till now described, and we obtain very good z-score in different matrices such as meat, feed, and offal. And now we can look to the future, because we can talk about the 7500 plus system that is very similar to the 7500 as regards selectivity and sensitivity. The method transfer is very simple and quick. We can see on the right, with 2 clicks, we can copy the mass method from the old system to the new system. But the novelty of this instrument is the robustness. So we have installed the 7500 plus system in our laboratory at the end of August, and but we don't have now a complete set of data in order to perform the comparison between the two systems. But we have a starting point, because we perform a preliminary test through the injection of 7 level of matrix matched calibration curve in liver extract, that is a very dirty matrix. These 7 level are injected for 29 times. So this results in more than 200 injection of matrix and more than 80 hours of analysis. These are the results, and they are good results because we can see the for some compound. I report some example. We have for each calibration point the 29 injection. So we have the mean, the standard deviation, and the percent CV, and we can see that the percentage standard deviation is very good, because in this case we have 2 macrolides is around 10%. Then we can see for another macrolide and beta lactams also very good value. For some compound, we have percentage CV between 10 and 15, but also in this case we have, for example, in this case sulfonamides, and in this case quinolones. But these results are very promising for the robustness of the new instruments. So, concluding, the developed LCMS method allow to analyze compound with very different physical chemical properties belonging to antibiotic classes in complex matrices such as meat samples. The LCMS triple quad 7500 gives the need selectivity and sensitivity for determination of antibiotics in meat samples. The use of matrix matched calibration curve allowed to obtain good recovery overcoming matrix effect due to the signal suppression. The LCMS method was validated in order to evaluate recovery and repeatability and calculate method uncertainty. The laboratory participation to proficiency test with very good results confirmed the good method performance. And finally, the method transfer from 7500 and to 7500 plus system, and the preliminary test seemed to indicate the same selectivity and sensitivity and very good promising robustness for complex matrices. Thank you for your attention. If you have any question, thank you.